Okay, so yeah, I'm going to talk about some work we did as part of the Rust program here in New Zealand with collaborators over in Australia and Queensland, particularly Australia and Swedish work and really important for this work. Yeah, so we've finished the last talk and we've kept it nice and cruisy and not complicated at all, right? Because we're going to talk about life cycles of rusts. And unfortunately, that's not the case because they're really, really complicated. So bear with me, guys. We'll get through it. Rusts have some of the most complicated life cycles of fungi, and they can produce up to 5 different spore stages and potentially alternate between 2 different completely taxonomically unrelated hosts. Australasian has 3 known spore stages that have been recorded. So we have the asexual stage, or uridinospores, that cause these sort of epidemics of disease or metaceae. And we know that the rust can produce telia or teliospores, and those teliospores germinate to produce the gametic spores, basidiospores. But there were some questions around whether or not those basidiospores infected an alternate host. So is there another host involved, or do they infect metaceae, and are they autologous and complete their life cycle on the same host? And the other spore stages have never been recorded in this pathogen. There was some work done prior to this study that we're talking about now, looking at whether basidiospores could infect metaceae. But often, or pretty much every time telia produce, the asexual spores are in there as well, right? So it produces uridinia inside along with those sexual spores. And so the work that was done prior to this point, we weren't sure whether it's contamination by those sonospores that would cause infection. But this work conclusively kind of showed that the sexual spore stage can infect metaceae by filtering those spores. So the basidiospores are really much smaller in size. They filter them out, infected Cyzanthemum, so that's the metaceae that we've heard about a few times today. And they started with rust that had a single genotype there represented up in P1, and they got infection, and then when they looked at the genotype of those rustosauri, they were recombinant. So you've gone from having one genotype present to all these different recombinant genotypes. So that's a pretty strong evidence there that we've had both infection by basidiospores and that meiosis and recombination have occurred as part of the sexual reproduction. So based on this, the authors of that paper put forward the following life cycle. So we have the clonal stage, the uridinia and we're all kind of familiar with the powdery yellow symptoms that you see. And then you can get the first part of the sexual life cycle, so teliospores. They produce these kind of more compact brown pustules on the leaves. They can then germinate to produce basidia and for basidiospores. And it's a bit hard to get a good photo of germinating telia, but this is kind of what they look like. You get these sort of white threads that come out of the brown pustules. And then this next part is kind of theoretical because no one's actually shown it. But the theory is that those basidiospores then infect the metaceous host, and you get anastomosis of the hyphae that's produced in plasmogamy. So you go from having haploid cells back to having a dikaryotic cell, and then from there, that dikaryote and then invariable to produce uridinia or telia again. So this is the current kind of theory around what the life cycle looks like based on that work. And we know that Australasian have been recorded in many countries around the world. And we have these different kind of genetic groups or strains associated with different hosts in its native range. And then we have these kind of invasive biotypes or strains that at the time have strong host associations all around different parts of the world. And Australasian in New Zealand is believed to have originated from Australia. So it's travelled to New Zealand in the wind, blown over here. And so some work done prior to that point and published in 2015 looked at genotypic diversity in Australian populations, and it's Myrtle et al. 2015. So they sampled quite a lot of isolates, probably just over 100 isolates, and they found that 99 of those were exactly the same. So There's a little bit of a few variants, but pretty much all the isolates they sampled had the same genotype. And so that was quite strong evidence that the population of the rust in Australia, at least, was primarily clonal. So the reproductive mode is clonal, and a little bit of variability that was detected is probably due to mutation. Similar kind of patterns in terms of wild clonal populations have been reported in Hawaii, Caledonia, and other invasive populations. And then some work was done looking at isolates from New Zealand. And so what we have here is a PCA. We've got some isolates from South Africa in the bottom left corner in green there, and then some isolates from the native range of at the top, and then the pandemic isolates cluster all down here in the bottom corner. All of the isolates that were sampled from Australia are all identical, so there's 20 different samples there, but they're all like right on top of each other. But you can see that there's 16 isolates from New Zealand, and they're not the same. They're kind of slightly different. So that was sort of interesting. It's like unexpected. We expect these invasive populations to be clonal, but this suggests that there's some diversity in New Zealand, even though it's come from a source population that's also expected to be clonal. And a little bit more work was done, some more sampling was done in South Africa. So this is just another way of representing this kind of diversity. So we've gone from 7 isolates to 40 something or whatever is in there. And they were compared to another small number of isolates from New Zealand. And again, we had this kind of variability. So like this sort of indicates that there's some genotypic variability in the New Zealand population as well as in the South African population. But the sampling effort for New Zealand was pretty low at this point. So what we wanted to do was have a better look and see, is there really some really strong evidence that there might be such a reproduction occurring in the New Zealand populations of this invasive rust. So we undertook sampling and monitoring in 9 locations, so you can see where those areas were. And we collected isolates over 2 years and for some genetic analysis, and just a shout out to Perry, because this is one of those studies that's only really possible because of their genomic work. So we have a genome that we can map back to, and that's actually made this work possible. It's really cool. Yeah, so we were looking for the reproductive stages, doing some monitoring work, and we also collected isolates for genetic work. The process of isolating the rust and looking at the DNA, we isolated individual pustules very, very carefully, and then extracted some DNA. And then because we have a very small amount of starting material, we then amplify by using a whole genome amplification method. The alternative would have been to isolate individual pustules and then try and use those to inoculate plants and build up spores. And to do that with 500 isolates would have been a huge amount of work. So this actually enabled us to kind of look at a huge number of isolates. We send all that DNA over to AGRF for sequencing and GBS analysis. So genotype complex sequencing basically chops up the DNA into different bits, and then sequences it, and you get a sort of representative data set of single nucleotide polymorphisms across the genome without having to sequence the entire genome, because we've already heard whole genome sequencing can be pretty expensive, and it wasn't possible to do that with the number of isolates that we wanted to look at. And then that data underwent some bioinformatic analysis. So AGRF had to do the first couple of steps with filtering and X3 quality and all that kind of stuff happens. And then Alistair got hold of the data and filtered it based on missing data and so on, and we did some analysis. So what did we find? Let's start by talking about observations of Telia. So in 8 of the 9 locations, we were able to find the sexual spore stage, but it's relatively uncommon compared to the asexual stage, the yellow spore stage that we're all so familiar with. So it's really quite hard to find them. You have to really go for them in New Zealand, but they are there. We also found that we only really found them on sort of the more mature expanded leaves, so we didn't find them on fruits and flowers and other really, really soft young new leaves that were just coming out with the disease was present, but not the sexual stage, only on the sort of more mature expanded leaves. And this figure kind of also illustrates that there's a bit of a seasonal pattern, right? So the bottom layer there kind of indicates our sampling effort. So we looked at lots of plants, and we didn't find symptoms on all of them, and then on a subset of those plants, we saw the clonal stage, Eurydinia, and across a wider window of time throughout the year. And then the sexual stage was found on a much narrower window and on a much smaller number of hosts. And And we typically only really saw telia at our sites during the peak of disease in New Zealand at those locations. So I think someone, maybe Rob, earlier showed a graph of my natural observations where some things kind of build up over time from October, and when they get to December, January, they just peak and then they start to come down again. And despite quite a lot of observations on Intracellular diffuser, for example, and quite a few observations on salt mining, although our sampling was kind of biased towards locomotus, and we mostly saw telia on locomotus hosts rather than with those other hosts. So there's possibly some kind of host associations. Okay, so this just gives you some numbers in terms of what we got back from the genetic analysis, because finding the sexual spore stage doesn't necessarily mean that sexual reproduction is occurring. Telia have been found in Queensland quite extensively, for example. So they found the sexual stage in Australia quite a lot, but there's not really evidence that sexual reproduction is occurring. So we need to kind of marry that up with what we're seeing in terms of the genetic data as well. So we mapped out a whole lot of SNPs, and then we filtered based on missing data to make sure that we had a data set that wasn't full of gaps. And we did some more analysis. And the first thing we asked was whether there's any population structure in our data set, right? Because we want to calculate some population genetic indices, and you can't do that on the entire data set if there is structure. You have to do it on each population individually. But what we found was that there's no real structure associated with either sampling year, with host species that the isolate was, that the disease is isolated from, or with location. So all of the kind of variants are found everywhere in some proportion. That meant that we could sort of apply these kind of statistical analyses to the entire data set rather than piece by piece. And we calculated nucleotide diversity and this statistic called genus D, and the numbers that came out were kind of consistent with what we'd expect to find in a founding population and a basic population with low starting diversity. So that makes sense. That's what we kind of expected to happen. So that's good. And we also looked at this additional analysis using a split tree network. So this is a little bit much taken, but it kind of mostly tells us the same story, right? So there's no real kind of obvious grouping of colors or shapes. Everything's kind of spread everywhere. The center of that network is highly articulated, and that's a real strong signal of recombination in the data set. So that's interesting. We also found that some of the isolates that we took, we sampled twice, so we had technical replicates, and those should technically be exactly the same, and they all come out next to each other in the tree. So that makes sense. That's really good. They're not quite exactly on top of each other because there's some missing data and stuff in the data set, so they don't come out exactly the same, but it's consistent with what we'd expect based on the analysis that we've done in the data set that we've ended up with. If we calculate the index of association, which is a measure of linkage to disequilibrium across all the SNPs in the data set, we get an output that supports that inheritance of SNPs is random. Okay, so if the population was strongly clonal, inheritance systems wouldn't be random, they'd inherit the same alleles every time. And we can also look at this chromosome level. So remember there's 18, so 19 and 20 are a bit weird, and just ignore those, I shouldn't really be on the figure, sorry. But if we look at the first, there's 18 chromosomes, and we look at the linkage between pairs of loci across the chromosomes, we see that there's this strong decay of linkages as those two loci get further apart. So that's what we'd expect. You have loci that are next to each other physically should have a high degree of linkage because it's harder for recombination to split them apart. But if you do have recombination occurring, you get this strong decay of that relationship. If there was a lot of only clonal reproduction, for example, we wouldn't see such a strong decay of linkage. So this is also supporting what we're asking in terms of whether there's sexual reproduction. And finally, we can also look at something called the inbreeding coefficient. So what we found was that the inbreeding coefficient was close to 0, but it is highly variable across life cycles of chromosomes. So that suggests, again, that there's this kind of intermediate scenario where you have both clonal reproduction and sexual reproduction occurring in these populations. So in conclusion, it's a little bit hard to interpret this stuff because it's not either strongly clonal or only sexual. It's kind of intermediate stuff. So you have to look at the data and kind of make a judgment based on what we're seeing. But we do have pretty strong conclusive evidence that there is sexual reproduction occurring in the New Zealand populations of this rust. We saw high genotypic diversity and no evidence of linkage among loci based on the index of association and we saw strong decay of linkage across loci on the chromosomes, no population structure, and we also found the sexual spore stages in those populations. And this is important. The implications for this are quite important. Genotypic diversity is linked to the adaptive potential of organisms, right? So the more variance we have in the population, the more baseline material there is for natural selection to work on, the more likely we're going to get adaptive processes occurring in these populations where the rust can adapt to new hosts or environment here in New Zealand, or it may adapt to the treatments that we sometimes want to use. So, for example, this really highlights the need to work very carefully with things like fungicides, use of rotations that we talked about earlier today, because the evidence suggests that we have sexually reproducible populations here in New Zealand, and they may be well able to adapt to the control tools that we're trying to use against them. So yeah, that's me. Acknowledgements and other people were involved in